Next, we're going to be looking at membrane transport, both passive and active. So we have to go back to that function of selective permeability for the plasma membrane. The plasma membrane is selectively permeable, but what does that mean? Well, it means some molecules can easily pass through the membrane and some don't. And there's things that determine whether or not a substance can passively permeate a membrane or go through a membrane. Well, lipid solubility is a big one, channels of appropriate size, and carrier proteins. What is meant by a lipid solubility of a substance? Why would that matter? Well, the membrane is lipid soluble, so lipids, if they're trying to get through the membrane, can easily pass. What about channels of appropriate size? Well, if there's an integral protein that has an open channel and that molecule fits, it can go in or out of the plasma membrane. And with carrier proteins, they are specific to a certain molecule, but if they exist for a molecule, that molecule with usually energy can go in or out of the membrane. But what's membrane transport and what's it have to do with passive and active processes? Well, some membrane transport's pretty easy. No real energy in the form of ATP is required. And others require ATP, or molecular currency, adenosine triphosphate. So there's two ways for substances to cross the membrane, and we have to look at more than if just a substance is lipid soluble or there's a channel or carrier for it. We have to look at the dynamics of the plasma membrane and what's going on when it comes to concentration. When it comes to concentration in passive processes, a substance is going to move from a high to low concentration, because it's easier for something to go from where there is more to where there is less. So it doesn't take a lot of energy currency, really doesn't take any at all, just the movement of atoms. With active processes, that takes energy in the form of ATP, so cellular currency, adenosine triphosphate. And we would see concentration changes in that molecules would travel from areas of low concentration to areas of high concentration. And kind of going against the grain or against that concentration gradient is going to take that extra energy. Here in this example, we see passive diffusion or a passive process. Passive processes occur when something doesn't need anything but the movement of molecules to power that process. Here, a drop of dye is added to the beaker. Initially, there's a lot of concentration right where that dye enters the beaker of water. Over time, without adding heat or stirring or shaking the beaker, that dye will spread from the center outward towards the sides of the beaker, and it'll do so till it's evenly distributed throughout. That's because that dye is going to have molecules in it that are moving, and those molecules, as they move, are going to help that dye spread out from a high concentration to the lower concentration of dye, which is in the other part of the beaker that doesn't yet have dye. So passive transport is what we're going to look at first. Passive transport includes simple diffusion, facilitated diffusion, and osmosis. Diffusion is both simple or facilitated. Simple diffusion is simple. We don't need a carrier or channel protein. But facilitated diffusion does need those things. It needs something to facilitate the movement, like a carrier integral protein or a channel integral protein. And osmosis is the movement of water, whereas the other two types, diffusion, both simple and facilitated, is the movement of solute. But no matter which type of passive membrane transport we're looking at, we're going to be moving substances from a high concentration to a low concentration area. And we're not going to talk too much about filtration, but it is a type of transport that is considered passive and usually utilizes pressure as opposed to concentration gradients. So let's look again at simple diffusion, but in more detail. With simple diffusion, the collision between molecules in the high concentration area causes them to be scattered into areas with less concentration. If this movement pushes molecules from a high concentration area across a membrane where there's low concentration, that's considered passive transport. So when a solute moves through the phospholipid bilayer from high to low concentration with no ATP expended, and it didn't need a channel carrier or any other type of integral protein, then that's simple diffusion. Because lipid-like molecules easily do this, they oftentimes can perform simple diffusion. But if you needed a channel or carrier protein, we'd actually be looking at something much closer to facilitated diffusion. Another way to talk about diffusion, or a molecule moving from high to low concentration, is to say that it's moving down or diffusing down its concentration gradient from an extracellular to an intracellular area. So here's another example of that. Here we have lipid-soluble solutes moving from a high concentration outside the cell to an area of low concentration inside the cell. And notice no channel or carrier protein is required. So this is a great example of simple diffusion. It's simple because we need nothing to facilitate this movement. And can I affect this diffusion rate? Could I speed that up? Could I make any type of diffusion faster? Well, yes. Temperature, particle size, and concentration gradient all play a role in diffusion rate. If you increase temperature, you make molecules move around faster, and if those molecules move around and bump into each other, they're more likely to get pushed into areas with lower concentrations, maybe across a membrane. With particle size, the smaller the particle, the faster it moves, and the more likely we are to see collisions between molecules and them moving from a high concentration to a low concentration area. And concentration gradient is really just talking about crowding. If you have an area with a ton of particles, because it's highly concentrated, when those molecules interact with each other, because it's so crowded in that high concentration area, they're even more likely to go through a membrane or a space or travel away to an area where there is a lower concentration. And that brings us to facilitated diffusion. Facilitated diffusion is really similar to simple diffusion, except we're going to bring in integral proteins, either channel or carrier integral proteins, because facilitated diffusion is facilitated by carriers and channels. And this is what we would use if we want to move something that's maybe not lipid soluble. Maybe it's a little bit bigger, maybe it has a charge, maybe something like glucose or amino acids or ions. And we still want to transport it passively. It's still moving from a high concentration to a low concentration, but it's hard to get through the membrane by itself. So facilitated diffusion is when solute moves through a transport protein from high to low concentration using a channel or carrier protein. Still no ATP expended, and the channels usually move mostly water and ions, and the carriers are a little bit more specific, and they move things like glucose and sucrose and fructose and amino acids. But facilitated diffusion, we use carrier proteins, is going to exhibit a certain amount of specificity, meaning that carrier proteins bind to specific things and nothing else. So that was what was meant earlier on when we talked about the fact that the plasma membrane is selectively permeable, and that you need a channel or carrier protein of a specific type to let some things through. Some channel proteins are always open, others are gated. So there's all of these dynamics that come into play besides just something being in greater concentration in one area and not in another. We're also going to be looking at the interplay between the movement of water as a solvent as opposed to the solute. Thus far, we've been looking at the movement of solute, of the particle that was dissolved in water. Next, we're going to be looking at water and its movement. Looking at water and its movement is osmosis. This is still a passive process, but we're not looking at the solute anymore. We're looking at the solvent. So osmosis is the movement of the solvent, usually water, across a selectively permeable membrane. And so water can diffuse through plasma membranes from high to low concentration. If you're never sure which way water is going to move, it's always going to move towards the area that has more solute. Because if it has more solute, it has less water. So water, when it passively moves by osmosis, still moves from areas of high water concentration to areas of low water concentration. But sometimes that's written as water moves by osmosis from areas of low solute concentration to areas of high solute concentration. But that implies the high to low water concentration movement. And things happen when water enters and leaves a cell. Cells, especially animal cells without a cell wall, can swell or shrink, but how they work because of this disruption in volume can be disrupted further to a point where the cells no longer function and can't retain or get back that functioning. So next we're going to talk more about osmosis and the diffusion of water across a membrane and terms used to describe osmosis and two solutions that are performing osmosis between each other, like hypotonic, hypertonic, and isotonic. So make sure to watch that video.